My name is Matt Hutchings. I work at the John Innes Centre, which is a research institute in Norwich. We research into plant health, microbiology, and how soil microbes affect plant health. And today we have a group of school children coming in to do some hands-on experiments, learn more about science and learn about why soil smells.
We're going to collect some soil. We just need a little bit, not a lot, just a little bit. Scoop it up with your falcon tube from anywhere.
So does that soil, does it smell?
Once done, pop lid on. You got your sample. Well done. Hands up if you've been in a lab before. You have. So it's people's first time in the lab. Yeah, that's really cool. So it's a nice sterile environment. We keep things nice and clean. So we're going to try and keep things nice and clean today, yeah?
So the bacteria that we work on are called Streptomyces, and they produce a compound, a chemical called Geosmin, which literally means earth smell. So Streptomyces bacteria and fungi make this volatile compound. So when it rains, it comes up into the air and you can smell it. And that's really what the smell of the soil is.
What you're going to do is you're going to scoop up a pea-sized bit of soil from this tube into your empty tube.
The bacteria that make Geosmin can't move, so they can't move around in the soil, they're just stuck where they are. So we think they make Geosmin, which is this earth-smelling compound, to attract insects and earthworms and other larger organisms that can actually pick them up, eat them, and spread them around through the soil. Because if they're stuck in one place, they use up all the nutrients. There's no food left, so they need to move to find new nutrients.
We've got our pea-sized amount of soil in our new tube, yep? We're going to add some water. We're going to do that using your little pipettes. What we're going to do now is spread our sample on the plate. So these are the spreaders. And what you're going to do is just simply spread. Go around in circles, make sure it covers every single bit of your plate, yeah? What's going to help the bacteria grow on this plate? What do we need to live? Oxygen, yeah. Air, it's got air, yeah. Yep, it needs food, so like nutrients to survive. That sounds good. Anything else? Water, yeah, perfect. So we've got hydration, we've got food, nutrients, everything on this plate to help the bacteria grow. Guys, you've done your experiment, so now that bacteria that was in the soil has all the nutrients, all the moisture, all the happiness it needs to grow. So now what we're going to do is we're going to put it in a warm place and it's going to grow over the next week or so, and it should look something like this. I think you've seen it all now. Hands up if you think this is gross.
My name is Kelly Rose O'Reilly. I'm a third-year PhD student at the John Innes Centre. I work with Streptomyces, a soil-dwelling bacteria that produces a lot of the antibiotics that we use in clinics today. I manipulate the Streptomyces to produce new antibiotics for us to use.
So almost all of the antibiotics we use come from soil bacteria and fungi. So penicillin comes from a fungus, things like streptomycin comes from soil bacteria like Streptomyces. So soil is the most precious resource on Earth because we need it to grow crops. Without soil, we can't grow any food. But it's also important because most of the antibiotics that we get come from soil microbes, and we hope that the next generation of antibiotics to treat drug-resistant infections will also come from soil bacteria.
So we're going to do a smelling activity because smelling is really important for us. If we smell something gross, we might not go near it, right? If you smell something really good, you might go to it, and bacteria do the same thing, where you're handed these little pots. Not going to take the foil off, we're just going to put it under our nose and have a little smell, and don't smell until I tell you. And what you're going to do is walk around the room, and it's important to do this in silence, so no talking. We're only going to communicate through smells. You can smell what other people have got, and when you find someone who's got the same smell as you, you're going to link arms, okay? And this is how bacteria might talk. They can't really say words to each other, can they? So they have to smell things. If we're in a group, let's cluster together and then separate out a bit. But what you can see is what's happened here, that bacteria, which are you guys, they've found others just like them and they group together, right? So you form in what we call colonies. So when Kelly showed you those pictures earlier and we saw little circles of bacteria, that's a colony. So you guys have formed colonies based on smell. It was really great to see the children's different reactions to the smells. So we provided them peppermint oil, we also had Geosmin from streptomyces and vinegar, and they had to communicate the different smells that they had through facial expressions, through exchanging of the different smells, and it was really interesting to see how that language developed without being able to speak.
It was great to have the children here today. We gave them hands-on experience of what it's like to be a scientist, and I think they learned why soil is so important to us as humans, important to our survival, and of course they learned why soil smells the way it does.